All right, guys. So in this lecture, we are going to look at heredity. Heredity. Now, in our previous lecture, we talked about the cell cycle and the cell division. Then we looked at the various phases that are involved in the cell cycle. We said that cell cycle consists of two main phases. We have the interphase and the mitotic phase. Now we said the interphase is the resting phase of the cell, where the cell grows and prepares for division. That is the mitotic phase. So the phase in which the cell actually grows and prepared to be divided is what we call the word interphase. It is the phase between two mitotic phases, hence the name inter. When you say something is inter, it has to be between two things. Is that okay? International, between two countries. Inter-household, between two or more houses. It's not necessarily between two, but at least between something and another. Are you getting it now? So that is that with the phases of the cell cycle. We said there are two phases where we talked about the interphase and then we talked about the mitotic phase. We say if we have a mitotic phase, then we are going to have an interphase, then mitotic. You can see that in between two mitotic phases, you have the interphase. This interphase is the resting phase of the cell. We all agreed on that in our last class, where we said that also the interphase is divided into three phases. In some classifications, they are actually divided into four, right? But basically, there are three phases. We have the G1 phase, which is called the first gap. Then we have the S phase, which is the synthetic phase. And then we have the G2 phase, which is the second gap. Now, we talked about the G1 phase. What are the activities that takes place in the G1 phase? Also, we talked about the S phase. What are the activities that takes place in the S phase? And then lastly, we talked about the G2 phase. What are those activities that takes place in G2 phase? We discussed all that in our previous lecture. Then we talked about the GO phase, which is also called the quiescent stage. Now, we said that there are some cells that do not actively divide. Are you getting it? They do not appear to exhibit division, e.g. the myocardial cells of the heart, that is the heart cells. And many other cells, they only divide occasionally as needed to replace cells that have been lost in our body due to injury or cell death. I also taught you in that class that there are two types of cell death that we can experience in the human body. The two types of cell death, we have apoptosis and necrosis. Where I told you that apoptosis is called the programmed cell death, and then necrosis is on a different one entirely. That is the non-programmed cell death. Now, the GO phase, let me explain again. In this phase, the animal do not appear to divide, or the cells do not actually exhibit division. So once it leaves the G1 phase, it goes straight into GO phase. It does not go from G1 to S phase to G2 and then to mitosis. Are you getting it? For this phase, what happens is once it leaves the G1 phase, it goes to GO, which is more like hibernation. You know when something hibernates, it's dormant. It's not actively like doing its activity. You were taught hibernation in ecology when you say a particular animal is still active. There are various processes going on in that animal's body, but it's just dormant there. Are you getting it? Same way applicable to the GO phase. That phase where the cell do not appear to what? To divide actively. Or in some cases, they only divide when there is cell death. That means person don't die, you will got to replace them. Are you getting it? That phase is what we call the GO phase. And only few tissues and cells in our body actually undergo this GO phase, of which one of them is the myocardial cell of the heart or the muscle cells of the heart. We say the heart is called the cardiac. Whenever you hear of anything cardiac, it has to do with the heart. Myocardial cells of the heart. You should take note of that. You guys are still in year one, so as time progresses, you are going to get acquainted with some of these things. We looked at all that. All right. Now, we talked about the mitotic stage. We said the mitotic stages, there are two. We have the karyokinesis, which is the main mitosis, and then we have cytokinesis, which is the division of the cytoplasm. Karyokinesis has to do with division of the nucleus. Cytokinesis has to do with what? The division of the cytoplasm. Is that okay? Cytokinesis, cytoplasm. Kinesis has to do with what? Division. Carrion. Anytime you hear of this carrion or you see carrion, it has to do with the nucleus. So when you hear of karyokinesis, they break down on the division of the nucleus. Cytokinesis, breakdown or division of the what? Cytoplasm. I told you kinesis, breakdown, carrion, nucleus, cytoplasm. These are the two mitotic phases. The karyokinesis is the main mitosis, while cytokinesis has to do with division of the cytoplasm. And then we outline the various stages: the prophase, the metaphase, then anaphase, telophase, and then we looked at cytokinesis, where the two daughter cells are formed. Is that okay? So that was what we looked at in our previous class. And we said that one of the clinical application of this branch of biology that you studied is in cancer. We said that cancer is a disease caused by uncontrolled cell division. We are talking about cell division. So when there are uncontrolled cell division, it tends to lead to cancer. There are genes that actually check or put this cell division in place to ensure that when there is need for division, the cell divides. And when there is need or there is no need for division, the cells stop dividing. Or we could actually say that there are genes that actually regulate how this cell divide to keep them in place or to checkmate the activities. If there is a fault with this gene, you will now have what we call uncontrolled cell division. And this uncontrolled cell division can form tumors, which we said becomes cancerous, and then they spread to other parts of the body via a process called metastasis. Although, if you have breast cancer, for instance, uncontrolled division in the breast, and it spreads to other parts of the body, like let's say kidney, you say it has metastasized, metastasis. Are you getting it? And that metastasis, because it spread to kidney, you are not going to say this is a kidney cancer. It is still breast cancer that metastasized to where? To kidney. Is that okay? So, and we said that the cancer is caused by what? Damage to genes that control cell cycle. I don't turn them before say these genes, and then they regulate what they go on in cell division to checkmate the activity of cells that are actively divided. Now, if this gene, there is an abnormality or abnormality with them, you see that the cell will divide uncontrollably, and too much of everything, of course, is bad, which leads to what we call cancer. These genes, told you, they are called oncogenes and the tumor suppressor gene. One actually aids in the cell divide, while the other one suppress what? This cancer or uncontrolled cell proliferation. Right? When I say proliferation, I mean say the thing they plenty, just they multiply. Are we getting it? So we looked at cancer. It's not something that is actually new to any one of you. This is just a brief breakdown of what we did in our previous class. Now, in today's class, we are going to look at heredity. Now, some of you, when we look at you and then look at your family lineage, there is tendency of us having something to like pointing to one person in the family and be like, you resemble this person. It might not even be your immediate parents. It might not be your sisters. It might be your grandparents. It might be your great-grandparents, or just random persons like that. And sometimes it might even be your mother's boyfriend. As a then, things happen. You understand? So, hereditary is the passing of traits or characteristics from parent to their offspring. Now, these traits may not necessarily be just physical traits, like what we can see physically. They could either be physical or behavioral traits. There are some of you that behave exactly like your parents, or exactly like one of your sisters. You talk like them, your behavior, your thought processes, everything. Are you getting it? So, when we talk about heredity, it's not just in the phenotype, that is the physical outlook of that person, not the physical appearance of that person. It goes beyond that. Are you getting it? But the one that most of us, we often look at is the physical appearance, which is the phenotype. So, hereditary is the passing of traits, characteristics, stored in the gene. Are you getting it? From parents to their children, which is their offspring. And these traits We can divide them, or we can include eye color. What are the colors of your eyes? Do you have dark eyes? Do you have brown eyes? Do you have blue eyes? Purple? Red? I've not seen purple or red eyes. Yes, we have seen red eyes. Those are smoky blue. But generally, the colors of eyes we have are black, brown, and bluish something. Now, this trait can include eye color. The type of hair is your hair. I don't know how to pronounce this thing, whether blonde or blonde or normal dark hair, and so on and so forth. What blood group are you? Are you A, B? Are you A? Are you O? Or blood group B? Are you getting it? What height, or where is your height? Are you short or you are tall? Sometimes when you look at your parents, you discover that your father is very tall. Your mother is not that tall. Then you are not that tall, like me. Are you getting it? At the end of the day, you might have carried the traits of height from your mom, and even the risk for certain diseases. Now, in the hospital setting, when we want to take, let's say, examine a patient, there's an aspect of that examination that has to do with family history. You understand? There is tendency of you having certain diseases, having certain illness or sicknesses because your parents or someone in your family has them. That is why you take family history. When you get to the hospital, maybe you're having symptoms of diabetes or hypertension or cancer. They will be like, is anybody in your family having this sickness, cancer? Or one of the sickness that they actually check family history very well is psychosis, schizophrenia, mental disorder. Man, put it that way, because those ones are big English. Mental disorder. That one, you have to do your normal mental state examination. How is the person? Is the person calm? Is he talking irrationally? Or after that one, they will not take family history. Is there anyone in your family who was mad, or who has been mad, or who is still mad up to now, or who have actually had this kind of sickness before? Same thing with hypertension. Same thing with diabetes. If you have diabetes or you are having symptoms of diabetes, when you get to the hospital, they will ask you, is there anybody in your family who have actually had diabetes? Same thing with cancer. You take family history. It's one of the most important thing you take when you are taking the data from your patient. Are you getting it? Now, heredity explains why children resemble their parents and why traits tends to run in family. And we said that the science of hereditary is called what? Genetics. You should take note of that. The science of hereditary is called what I just said, genetics. Is that okay? Science of hereditary is called genetics. Now, let us look at some key terms in hereditary. One of the terms that we are going to look at is gene. A unit of hereditary, or a segment of the DNA that controls a particular trait, is called a gene. For instance, you will say, this gene dey your body. When you hear people, they say things like, this gene dey your body. What are some of the gene you can say dey your body? You say your papa dey drink ogogoro. You now, you still carry ogogoro, begin to drink. You say, this gene dey your body. Or maybe you like fair, your dad married a fair woman. And then you, at the end of the day, you discover that you are still liking fair women. You say, this gene dey your body. What do we mean? In the Bible, it was like that. You understand? If you look at the story of Abraham and Sarah, Abraham was like, Sarah was fair to look upon. Then Jacob, Isaac, the son they gave birth to, Isaac also went to a new land and looked for a woman, I think Rebecca also, who is also fair to look upon. So they have the trait of liking fair women in their family. It's a gene. So a unit of hereditary is a gene. Now, a segment of the DNA. I taught you earlier that the DNA contains nitrogenous nucleotides, right? Which are arranged, joined together by phosphodiester bond. We have looked at all that. If this is a DNA molecule, right? Right at the helix. And then we say adenine must bind with what? Thymine. Cytosine must bind with what? Guanine. A segment of the DNA that actually regulates a particular trait is called a gene. So it may be the trait for brown eyes. It may be the trait for your color of hair. Are you getting it? That is why if you have, let us say, somebody who is kind of half caste, you see them, their skin may definitely be like those of the white, but their hair is still looking more of Nigerian. Probably they inherit the gene for their skin from the parent who is a white, and then inherit the gene for their hair from the parent who is a black person or an African. Is that okay? So a segment of the DNA that controls a particular trait. What are the traits we've talked about? Height. That segment of the DNA that controls height is what we call a gene. Is that okay? Exactly. So that is that. Let's look at the next one, the chromosome. I have taught you extensively on what chromosome is, right? Now, we said that a chromosome is made up of DNA that contain many genes. So when you have many genes, you call it what? A chromosome. I have also told you that the chromosome consists of DNA plus protein. What kind of protein? Did we learn? Told you that it contains histone proteins. Histone proteins. Are you getting it? And we said we looked at the structure of the chromosome. We actually dealt deeply on that. So a structure made up of DNA that contains many genes is called a chromosome. I've said that humans have 46 chromosomes, which are in pair, 23 pairs. We looked at all that. When I say 23 pairs, it means 23 into 2, which gives us what? 46. Of which this 23 pair, a pair is the sex chromosome, and the other 22 pairs are autosomes. We looked at that. The sex chromosomes are the one that regulates the sex of the individual, whether the person should be a male if it has XY, or the person should be a female if it has XX. Is that okay? A structure made up of DNA per protein that contain many genes. We've said that a gene is just a segment of the DNA that controls a particular trait. So when you have a structure that is made up of DNA that contains as many genes as possible, you call that what? Chromosome. And we said there are 46 what? Chromosomes. Now the DNA, the molecules that carry genetic information, deoxyribonucleic acid is a nucleotide, or a polynucleotide rather. The DNA is a polynucleotide. I have taught you about DNA before, so I wouldn't really delve deep into DNA. It's a polynucleotide. Then allele. I don't know how they actually pronounce this word, whether allele or allele or Allah. They seem somehow anyway. Allele. The different versions of a gene is called allele. For eye color, is it blue or brown? The different versions of a gene is called allele. What do we mean by that? For eye color, does the person have blue eyes or brown eyes or even dark eyes? Does the person, is it tall or short? Biology textbook, your secondary school biology, modern biology, say the alternative forms of gene is called an allele. The alternative forms of gene is called an allele. So when you look at somebody, there are two alleles that are actually possible, or depending on the particular trait in question. For instance, you can either be tall or what? Short. That's an allele. Are you getting it? Then for a gene that controls blood grouping, you can either be AB, AA, or what? O. That is what? An allele. The different versions of a particular gene is called the allele. Are you getting it now? For instance, this is an allele, tall or short, blue or brown, AS, AA. Are you getting it now? All that. So that is what the allele is all about, the different versions of a gene that controls a particular trait. Now, genotype. Genotype is the genetic makeup of the organism. What the gene says is what is present in the genotype. The DNA, the sequence of the DNA. What proteins are there? How are they arranged? What is the nucleotide sequence? The genotype. It's more like the genetic makeup of that individual. How many of those genes are collected from the father? How many are gotten from the mother? Joined together to make up your own genetic makeup. Are you getting it now? So the genetic makeup is what the gene says, what characteristics are being expressed physically by virtue of what is inside. So the genetic makeup is what is inside, while the phenotype is what is outside, expressed by virtue of the activities of the genotype inside. Do you get it? Genotype is what is inside. What are those genes you've inherited? Then the phenotype is the expression. So genotype gives expression for phenotype. Are you getting it now? For instance, I have a gene for, let's say, medium-sized height. Is what is inside of me. But the expression will be, what height are you seeing? Do you get it? Exactly. So that is what the genotype is all about. I might have a gene for dark hair. The thing is, what are you seeing? You are seeing dark hair. So there are some genes that are dominant, and there are some that are recessive anyway. Are you getting it? But genetic makeup is more like what the gene says. What sequence of genes are arranged in your body? How many did you collect from your mom? How many from your dad? Are you getting it now? That's why it's sometimes when you say you want to go and do DNA test, the genotype and the rest. Now, DNA genotyping. Now, the phenotype, the physical appearance or expression of the gene. What can you see? Do you get the difference now? Genotype, what is inside? Phenotype, what can you see? The physical expression of those genotype. Is that okay? Exactly. It's more like your computer, your printer. Your printer works behind the scene to print something. There's something actually working. But when you come out, you see different colors. You see ink written on paper. That is the expression of what has gone on inside of that printer. Do you get the idea now? So that is the differences between all these terms we've looked at so far. We've talked about the chromosome, talked about the gene. We say a unit of heredity, a segment of the DNA that controls a particular trait. Then chromosome, talked about chromosome before. DNA, talked about DNA. The allele, say different versions of a gene. For eye color, is it blue or brown? Are you getting it? For height, is it tall or short? The gene could either be a tall gene or what? A short gene. Are you getting it? Exactly. For instance, this is an allele. This person expression, they are pairs anyway. But this one is for a tall one because the tall one will be dominant. Then this one is short one because, of course, that will be dominant. All right. So that is that. Now, homozygous rather. Homozygous, when you have two identical alleles for a gene, two identical alleles for a gene. This alternative form is an allele, tall or short. This is an allele. Are you getting it? Either it's tall or short. Is that okay? These two, they are the same. So this is not an allele. You should take note of the differences. Maybe the explanation I made earlier might want to confuse you. That's why I have to come back. This tall, short is an allele, alternative form of gene. Short is not an allele because they are two same gene. Is that okay? Now, coming here, homozygous having two identical allele for a gene. E.g., if you have tall or short, this is a homozygous allele, two identical allele for a gene. You have two, one for tall, another one for tall. This one for short, another one for short. That means if they are the same, you say is what? Homozygous. And homo have to be like the same. While heterozygous, having two different allele for a particular gene. For instance, you have an alternative form like this. This is for tall, and this is what? Short. Are you getting it? This is for tall, and this is for short. These two different allele for a particular gene. Two different allele for a particular gene. So that is that. Now, the dominance. There are some genes that express themselves even if only one copy is present. That one is called a dominant gene. Any gene that expresses itself if only one is present is called a dominant gene. For instance, if you have tau, this you can see this big T here. It will express itself. This small one is recessive. So any allele that expresses itself, even if only one copy is present, is called a dominant gene. Why recessive? An allele that only shows up when two copies are present is recessive. For instance, T. This T, there are two copies present. That is the only time you say the person is short. For instance, I might have something like TT, TT, TT. Now, among these three things you see here, only this person is short. When you have two short. So this allele is recessive. This allele here is what? Is dominant. T is dominant. T is dominant. T is dominant. But this small t here is what? Recessive. So big T is a dominant allele. Because when you are talking about the allele, we are not talking about a pair. We are talking about one of these T. So if I have TT, 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 this big T here is dominant. This big T here is dominant. This small t here is what? Recessive. Because it will only show up that this person is short if only you have two copies of this allele present. Are you getting it? This is the only short. If you learned your genotype hereditary in your secondary school, you'll be taught that this is the only short one you have here. This person is tall, this one is tall, this one is what? Short. So that's the difference between the dominant gene and recessive. Dominant, they express itself even when it is only one copy that is present. Even when you have just one copy, they express themselves. Like I have said, that this big T here is a dominant allele. Then for recessive, they only show up when two copies are present. Are you getting it? Like this small t and this small t is what? Is recessive. Is that okay? Now, so if you have, okay, let me not spend so much time here. Now, the basic principle of hereditary, the Mendelian genetics. Now, the first person that actually talked about the hereditary was Gregor Mendel, and he was known as the father of genetics. So you may be asked, who is known as the father of genetics? The correct answer is who? Gregor Mendel. He studied pea plants and came up with laws that still applies today. Now, it's a question they will ask you. What did Gregor Mendel study? He studied what? Pea plants, and came up with the laws that still applies today. There are two laws of Mendelian genetics. The two laws are, we have the law of segregation of genes. Now, the law of segregation of genes says that each parent has two copies of each gene, but passes only one copy to their offspring. You know, each parent have 46 chromosomes. We say they are diploid. Are you getting it? But at the end of the day, they will only pass how many? 23 chromosomes to the children. So you see, every parent have two copies of each of the gene, but we only pass only one copy to their offspring. Is that understood? Each parent has how many? Two copies, but we only pass only one to their offspring. So if you have, let's say, a parent that have TT and another one TT, like this parent you see here, we only pass one T from this one to form a child. This one bring one, this one bring one to give an offspring here. Are you getting it? That is what the Mendelian law of segregation of genes. If these are two parents, capital letter T, and then this one is T. This is gene. This T here will bring one, this parent will bring one, another parent will bring one, and then this one will give birth to two children. We give birth to a child who is very tall. Are you getting it? This one, the father, let's say this is a father. The father is tall. The mother is also tall. But the mother has a recessive gene for shortness. This one is not expressed, like I told you. It is inside. It is not expressed. Is that okay? This shortness here is not expressed. It's on the inside. So when these two people marry, the father will bring his tallness, the mother will bring his tallness for the first child, and that first child will be having all tall traits or all tall gene. Now, how about this one? This one, the same thing. They will also give birth to all tall. Then this particular one will come together, and then it will also be tall, but one of the child will now have a recessive gene that is what? Short inside. This one also will also come together, and then it will also give us something like this. So you see, from the process of passing on the chromosome, it is only one gene that the parents are actually giving out. This is what we call the law of segregation of the gene. When you say segregate, to what? Separate, to put apart. Are you getting it? The law of segregation of gene by who? Mendel, Gregor Mendel, says that each parent has 2 copies of gene, but passes only 1 copy to their offspring. Is that understood? Now, the next one, law of independent assortment. Now, the genes for different traits are inherited independently of one another. That is the law of independent assortment. When you say independent, they pick different ones. Are you getting it? You know me say you pick tall gene for this particular trait, then the other traits must be brown eyes. So you now say tall gene, brown eyes, let's say fair skin. Are you getting it? If you can pick a tall gene from one of the parents, then the eyes may be blue eyes instead of brown eyes. Then the skin may actually be what? Dark. That is what it's trying to tell you. It's not trying to tell you that for the fact that you pick a particular gene, it means the other gene for the other traits must be complementary to the one you picked. Each of these genes are independently collected or independently assorted. Hence, the law of independent assortment. Let me say it again. When you talk about the law of independent assortment, this is what we mean. Now listen. You know, it is more like there is no particular order of selecting the gene. Let us say you have an African mom. What are the characteristics of an African woman? The African woman will have normal dark hair, melanin skin, let's say dark skin, right? Let's say she's tall and just like this. These are various characters. And let's say her genotype AB. Now, the father, American man. Let's say the American man is fair. His hair is blonde, normal American hair. How do they call them? Blonde also. Then he's short, he's not tall. The blood group, let's say O. Now, if these two people marry, what we are trying to tell you is that for genes, this one will bring, this one will bring. What we want to collect, we know me say for the fact say we collect darkness from mama, me say we will still collect dark hair, the tallness, and the blood group. As you collect the dark hair, at the end of the day, you might collect gene for what? Shortness. And then blood group what? AB. And this one, the skin may actually be what? I mean, the hair may be blonde, the skin may be dark, and then you have shortness and blood group AB. There is no particular order that, oh, as far as say the body they fair, the hair must be blonde, the the height must, the person must be short, or blood group must be O. No. Each of these genes are what? Selected independently of one another. So that is why when you see somebody, it's very, most times it's very, very difficult for you to see. There are some persons that actually carry the trait of everything. When you look at the person, it's more like a photocopy of their father. But when you look deep down, you see some of the traits of the mother in that person. Are you getting it? Now, this process is a randomized process. So there are tendencies that the possibility of them collecting traits for only mothers is very, very high, but is still very, very low. Are you getting it? It's a randomized process. It's not a process that is programmed to say, okay, this we collect 75% from this. So that is the law of independent assortment. Say that genes for different traits are inherited independently of one another. Then the law of segregation of gene, each parent has 2 copies of each gene, but passes 1 copy to another. Are you getting it? Exactly. So now we have dominant and recessive inheritance. I've talked about dominant traits. Only one dominant allele is needed to show the trait. Such a trait is a dominant trait. For instance, tongue rolling. There are some persons that can roll their tongue, while there are some that cannot actually roll their tongue. For instance, me, I can roll my tongue. You can see it. Ability to roll the tongue. Are you getting it? That one is a dominant trait. It's either you know how to roll your tongue or you don't know how to roll your tongue. Are you getting it? I can give birth to a child today who does not know how to roll their tongue. Also, ability to taste phenylcobamine. It's a very bitter something. Are you getting it? There are some persons that when they taste it, they feel the bitterness. Why there are some that don't feel it? Do you understand? So these are dominant traits. So only one dominant allele is needed to show the trait. Once you have the dominant allele, let's say the ability to roll the tongue is arrow. You have big arrow, small arrow. So long you have this big arrow, you must show that trait. Are you getting it? Now, then recessive trait needs 2 recessive alleles to be seen. Inability to roll the tongue. So if ability to roll the tongue, you need just 1 dominant allele, tongue rolling. Recessive, you need 2 recessive alleles to be seen. For instance, the inability to roll the tongue. Are you getting it? There are some persons that they don't know how to roll the tongue. So for that one, you have 2 small arrows. You must have these 2 small arrows for you not to be able to roll your tongue. But once you have 1 big arrow, you should be able to what? Roll your tongue. Those are the 2 types of inheritance. Dominant trait, recessive trait. All right? Exactly. Now, there's what we call codominant. Now, codominant, both alleles are equally expressed in the phenotype, e.g., blood group AB. Both alleles are equally expressed in the phenotype. That is, you see them. For instance, A is a blood group, B is a blood group. But in a situation where A and B combine, then you are having what? AB. Are you getting it? A is an allele, B is an allele. Is that okay? But when you now see AB, that means the 2, allele A and B, are both what? Expressed in the phenotype. What you see is AB. Do you get the idea? I say A is an allele, B is what? An allele. Normally, if it is normal, all these heights, tall and short, ability to roll the tongue and inability to roll the tongue, you will discover that either this dominant one will be prevalent or ability to roll the tongue will be what? Prevalent. But for codominance, 2 of them, they are dominating together. So A will be expressed in equal amount, B will be what? Expressed in equal amount. Hence, AB. That's why we say AB is a universal recipient. Are you getting it? Blood group A can donate to it because it's expressed. Blood group B can also what? Donate to it. Do you get the idea now? So both alleles are equally expressed in the phenotype, in the physical outlook. Blood group AB. There is no one that is dominant and the other one is what? Recessive. If, for instance, blood group A is dominant, it means you should not be able to donate blood group B to AB. Why? Because A is dominant, hence B will be what? Antigen. Are you getting it? Exactly. So that is the general idea for codominance. As name implies, codominant, we are living co-dominant, living together. Cohabit, living together. Are you getting the idea? Exactly. So that is the idea of codominance. Incomplete dominance. Now, for incomplete dominance, neither of them is completely dominant. The result is a blend. It happens. For instance, when you have red flower and white flowers come together and they are forming pink flower, that is incomplete dominance. All these things are things they usually set in your JAM. Codominant, incomplete dominance. A situation where red flowers and white flowers come together and then they are giving pink flowers. This one bring part, this one bring part, they come together, come from pink. That one is what? Incomplete dominance. Is that okay? Exactly. Had it been one is completely dominant, maybe red is dominant, it's only red you will see. If white is dominant, it means it's only white you will see. If both of them, they co-dominate, like co-dominance, you will see red and what? White in the new offspring. But the situation where red and white come together and then it form pink, then that one is called what? Incomplete dominance. They usually set this in JAM when they will ask you when red flowers and white flowers come together to form pink. This is also known as codominance, incomplete dominance, then dominant and recessive. You should be able to distinguish between them. All these ones, they are types of inheritance. We have dominant inheritance, recessive inheritance, codominance, incomplete dominance, and then we have what we call sex-linked inheritance. Sex-linked inheritance. Now, these traits, they are linked to sex chromosome, especially the S chromosome. These are called sex-linked inheritance. Now, my dear student, for instance, I told you that we have 23 pairs of chromosomes. Did we learn something like that? 23 pairs. Out of the 23, 22 are autosomes, the one that regulates other functions in other traits in the body. One pair is a sex chromosome, right? And the sex chromosomes, we have XY, and then we have XX. Now, there are some traits that are especially linked to the sex chromosome that if you can only find them in the sex chromosome, for instance, the X chromosome, e.g., color blindness and hemophilia. These are called sex-linked traits. Traits linked to only sex chromosome, especially the S chromosome. Now, we are going to see the clinical or the significance of this XY chromosome as time progresses. We say e.g., color blindness and hemophilia. All right. Now, like I said, humans have 23 pairs of chromosomes. 22 pairs are called the autosomes, and one pair is the sex chromosome. For female, the chromosomes, we have the pair XX. While for male, we have the pair XY. Now, the sperm determines the sex of a baby. If the sperm carries an X chromosome, you know the woman only have XX. So it is the sperm that will tell us whether the child will become a girl or a boy. So if the sperm carries S chromosome, automatically the lady will bring only X. So S combined with X, it gives a female child, a girl child. However, if it carries Y chromosome, the X from the female will combine with the Y from the man to form what? XY, which will give a male, a boy. Is that okay? Now, so that is that for the chromosome and sex determination. I have actually explained this before. Why did I have to bring it back? But then we just revised it now. Is that okay? Now let us look at some genetic disorder. Genetic disorders, they are health conditions caused by problems in the genes or chromosome. They are health disorders or health conditions caused by problems in the genes or chromosome. If there is chromosomal aberration, when I talk about chromosomal aberration, we are talking about abnormality in the chromosome will cause serious health conditions. And these health conditions, they are called genetic because they have to do with what? An aberration of the gene. So we call them genetic disorder. Examples include sickle cell anemia, Down syndrome, and hemophilia. I have actually looked at Down syndrome, where we said Down syndrome is trisomy X. But we'll still discuss it a bit here. but we've looked at Down syndrome earlier. So these are health conditions caused by problems in genes or chromosomes. Talked about sickle cell anemia, Down syndrome, and hemophilia. Now, what is sickle cell anemia? Sickle cell anemia is a genetic disorder caused by a change in the gene that makes hemoglobin. Hemoglobin is a protein in the body that carries oxygen throughout the body. Is that okay? A protein in the body that carries oxygen through the body is called what? Hemoglobin. Hem has to do with blood. Globin has to do with what? Proteins. Is that okay? So, sickle cell anemia is a genetic or blood disorder caused by a change or a mutation or an aberration in the gene that makes hemoglobin, the proteins in red blood cells that carries oxygen throughout the body. All right? Now, in people with this disease, what actually happens? The blood cells, specifically, we have different blood cells. The red blood cell, they are supposed to look like donuts or puff-like. They become sickle shape or crescent shape. Sickle in the sense that, sickle now, used for harvesting mango and the rest from tree in agriculture. It becomes sickle-like. Are you getting it? And these sickle-shaped red blood cells, they don't easily move like that. So at the end of the day, they will actually pack themselves together and prevent blood flow. You can see it. Prevent movement. So, and if they don't go to where they are supposed to go to and then supply oxygen, what do you think will happen? The person will lack oxygen in the body. Are you getting it? If you look at it, this is a normal red blood cell here. This is a normal hemoglobin here. Now, this is an abnormal hemoglobin here. Are you getting it? This is an abnormal blood cell. You see how it looks like a sickle shape. Is that okay? At the end of the day, what do you think will actually happen? You see how they are packed here, preventing movement through the blood vessel. And if there's no movement through the blood vessel, what are you expecting? Because they carry oxygen. And this oxygen is what is needed for metabolism and development in the body. If oxygen does not get to body cells, what will happen? And the person will choke and die. Or will have serious health condition. Is that okay? So that is what happens in sickle cell. It has to do with the red blood cell. They become sickle cell, preventing them from movement, or crescent shape. Crescent shape or sickle cell, sickle shaped. And then instead of the normal round and flexible disc shape, making it harder for the cells to move throughout the blood cell, you call that sickle cell anemia. Are you getting it? And it actually has to do starting from protein. But most of you may not really understand this till you get to 200 level when you have been taught protein. They will teach you the primary structure of protein. Primary structure of protein has to do with the sequence of amino acid in the protein. What do I mean by sequence of amino acid? Now, amino acid makes protein. Amino acid like glutamic acid, valine, and so on and so forth. Now, what happens in sickle cell anemia is there's supposed to be glutamic acid, which is water soluble. It's an acid, it's water soluble. This glutamic acid is replaced with valine, which is hydrophobic. That means they like water. So glutamic acid is replaced with valine. That alters the entire process, which causes what? Sickle cell anemia. But the link between these, you will not really understand it until when you come to 200 level, then you start learning about protein, the primary structure of protein, and so on and so forth. But just know that it has to do with what? A substitution in one of the amino acids. Are you getting it? The gene that codes for that particular amino acid is substituted. Instead of a gene that codes for glutamic acid, you are having a gene that codes for what? Valine. Do you get the idea? But that will not concern you now. The most important thing when you go know is say sickle cell anemia are genetic or blood disorders caused by a change in the gene that makes what? Hemoglobin. The protein, the red blood cells that carry oxygen throughout the body. Are you getting it? And we said what happens is that people that have sickle cell anemia, their red blood cells, they become crescent shape or sickle cell shape, thereby preventing them from moving freely. And because of that, these red blood cells, they will actually clump in one part of the blood vessels, thereby preventing oxygen supply to various parts of the body, which could cause hypoxia. Hypoxia has to do with deficient in oxygen level. Is that okay? And hypoxia can actually cause necrosis. What do I mean? Hypoxia can cause cell death. And if the cell is the basic unit of the human body. If there's cell death, what do you think will happen? The person will start having pain, sicknesses, this and that. Sometimes it grabs to breathe, and before you know it, you see those common characteristic symptoms of somebody who is an SS. Today you don't sick, tomorrow you don't sick. You most times when you rush to the hospital, you see them breathing, trying to breathe. Why? Because there is what? Low oxygen. So there's your body now wants oxygen demand. That's why when most times when they rush them to hospital, the first thing they do is to want to fix oxygen in them so that at least they will do that. But at the end of the day, nothing still come out. Are you getting it? So that is that for sickle cell anemia. Now let us look at the next one, hemophilia. Now hemophilia is a genetic bleeding disorder in which the blood doesn't clot properly. When you have a minor cut in your body, it is expected that after all you have a cut in your body, maybe minor or major, it's expected that after some time, your body mechanism or your body devises a mechanism that actually puts a clot. When we talk clot, we know you talk about wrapper, a layer of cells on top of that blood, or on top of that clot, that will prevent you from bleeding to death. Are you getting it? Now, the same most of accident victims, the reason why most of them die is because of what? Uncontrolled bleeding. They bleed too much and then bleed to death. Sometimes even gunshots is uncontrolled bleeding. They bleed so much and they bleed to death. Are you getting it? So hemophilia, now this is a sickness of its own. It's a genetic bleeding disorder in which the blood doesn't clot properly. And if the blood doesn't clot properly, what happens? You bleed to death. This leads to prolonged bleeding, even from minor injuries. Since when we say normally, your body will be just like, you will bleed. And sometimes even spontaneous internal bleeding, especially in joints and muscle. Yes, some persons that they have internal bleeding. And internal bleeding is very dangerous because you won't know that you're actually bleeding entirely. Before you see, you are gone. It's not like the physical bleeding that you can see and say, this person is actually bleeding. Now, when you get injured, your blood uses special protein. If I say when you get injured, your blood uses special proteins. These proteins are called clotting factors. Is that okay? Your blood uses what? Special proteins called what? Clotting factors. These clotting factors are numbered from 1 to 8 to stop bleeding. These factors work in sequence like dominoes, forming a clot to seal the injury. When we say they are working in sequence, like after 1 don't work finish, 2 go continue, 3 go continue, 4 go continue in that sequence. So if, let us say, you have 1 don't work, 2 don't work, and then it continues like that. Let us say number 5 can't get problem. The entire clotting process is altered and is destroyed. Hence, you see the person bleeding. Or let us say number 7 have issue. Did I say to 8? XIII. That is 13. Not 8. 8 is VII. This 13. They are numbered from number 1 to 13. So if 8 has issue, there's a problem. If 9 has issue, there's a what? A problem. All 13 of them must work in order for us to what? To have the blood, the clots that will actually prevent bleeding. Are you getting it? So that is that. Now, what happens in a hemophilia? One of these clotting factors is missing or doesn't work properly. So the clotting process is either slow or incomplete. That's what happens in hemophilia. I've told you that we have 1 to what? To 13. And they work in such a way that as this one work finish, you transfer the work beyond. That is how they work, and then they form that covering on top of your wound. When you have a wound, you discover that after some time you see a crust on top of that wound trying to heal. That crust is what is preventing bleeding. Immediately you remove that thing, you start bleeding again. Are you getting it? That is what we call the clot. So if that clotting process, maybe one of these clotting factors, they are proteins. These proteins are called clotting factors. If one of the clotting factors is having issue, that means maybe it is not working properly. The clotting process is slow or incomplete. Are you getting it? Because it's more like one cannot substitute for another because we said they are working hand in hand with each other. Now, as a result, bleeding lasts longer than normal. Bleeding lasts longer than what? Than normal as a result of that. And the types of hemophilia. Now, hemophilia is divided into two types. We have hemophilia A and we have hemophilia B. Hemophilia 8, what clotting factor, what clotting factor is missing? Clotting factor 8 is missing. Is that okay? For hemophilia A, clotting factor A, number 8 is missing. Then for hemophilia B, clotting factor 9 is missing. And the genetic cause is mutation in the F8 gene or mutation in the F9 gene. Is that okay? Exactly. Is that okay? Factor 8 is missing for hemophilia A. And there is mutation in the F8 gene. That means the gene that regulates factor 8, there is change. There is an abnormality. There is an aberration in that gene. Then we have hemophilia B. Factor 9 gene is missing. And because of what? Aberration or abnormality in the F9 gene. Now, which of them is more common? Hemophilia A is more common than hemophilia B. You should know that. Hemophilia A is more common than what? Than B. Is that okay? Then, hemophilia is S-linked recessive. I'm going to ask you this question. So please take note of it. Hemophilia is X-linked recessive. The gene for clotting factor 8 and 9 are located on the S chromosome. They are located in which of the chromosome? S chromosome. Now, male have one X and one Y chromosome. So if their S chromosome has the faulty gene, they will have hemophilia. Take note. Male have one S chromosome and one Y chromosome. Hemophilia is sex-linked. It's linked to the what? The sex chromosome. Which of the chromosome? The X of the sex chromosome. Men have one X. Women have one Y. Men have one X, one Y. Women have two X. Now, what happens is, if a man has this faulty gene of hemophilia in his X chromosome, there is no other chromosome to actually combat it. Are you getting it now? So that man will express what? Hemophilia. Do you get it? But female, they have two X chromosomes, XX. So even if one X has the faulty gene, the other one can compensate. They usually become carriers without symptoms, unless the two X chromosomes have the hemophilia. Then you know that, oh, problem don't come. But hemophilia, men are usually, they are the one that usually experience this excessive bleeding. If women experience it, and let's say a woman has hemophilia, that woman, during childbirth, it will be very difficult for her. She may not even survive childbirth because of postpartum hemorrhage. You go bleed before you see Otilo. Are you getting it? So, and postpartum hemorrhage, that one is another thing of its own. But during the childbirth, there are a lot of tear, a lot of bleeding from different places, and now women, I feel explain that one for now very well. Are you getting it? But generally, now male, now they get hemophilia. Do you understand? So male have X and Y, one Y chromosome. So if their X chromosome is faulty, they will have hemophilia. But female has two X chromosomes. So even if one has the 40 gene, the other one can compensate. They usually become carriers without symptoms. So women, they are carriers, they don't show the symptoms. Now, one thing that you should know is S-linked recessive. S-linked recessive in the sense that if only that one is present, are you getting it? And it's showing, if only it is present, that is the chromosome now, only X, only one is present, and it is showing, you say it's S-linked what? Recessive. Are you getting it? Exactly. So that is that for hemophilia. Now, if a carrier female and a normal male have children, what are you expecting? Now, this female is what? A carrier. Why the man is normal? You know, this is the only gene that can have hemophilia, the X chromosome, the X. That's the reason why you see that the Y will not put either small h or big H. The big H means the person does not have hemophilia. The small h means the person has what? Hemophilia. Is that okay? Now, if crossing takes place, what do you think will happen? The person will give birth to a female that is normal, is not a carrier. Now, it will give birth to a male that is normal, that is not a carrier. Are you getting it? Then it will give birth to one female that is a carrier, that she's a carrier. Is that okay? And give birth to one hemophilic male. One of these male. This is the only person who is hemophilic among these people here. This person here. Because the only X chromosome present is hemophilic. This big H means it's normal. It's not hemophilic. Small h means what? Hemophilic. Are you getting the idea now? So that at the end of the day, this one, female will be normal, not a carrier. This male will be what? Will be not normal. I mean, will be normal too. This is a male, normal. Then this female will be a carrier. And then you have one male that is hemophilic. So the question will now be, how many of the children will be hemophilic? One. Only one is hemophilic. How many are carriers? How many are carriers? Only one person is a carrier. The carrier is this person here. This female here is a carrier. You understand? How many of them are normal? Two of them are normal. These two. So for normal one, you have two. Is that okay? That is it. So that is that for hemophilia. Now, you are to answer these questions, 20 questions, and then you send it across to me on Monday. These are your CA questions for the week. For week 4, I didn't give you for last week. So these are your CA questions. Put them on a foolscap paper. Write your name, your department, and your serial number. If you forget to write your serial number, I'm not marking. Your name, your department, and your serial number. The serial number is on that score sheet I usually send to the group. Please, drop your pen now. Go and get the book you want to use to, or the foolscap paper you want to use to answer the assignment. Write your serial number first. Your name and your department. Because without the serial number, I'm not going to mark. Now, you have the first question. Which of the following is the basic unit of heredity? Is it DNA? Is it chromosome? Is it the gene or protein? I've told you. Let me answer this one for you. And the basic unit of heredity, you will see it in the first lecture, the first page of this lecture. The basic unit of heredity is in the first, look at it. See the answer here. So you go through all of them. There are about 20 questions here, or 19 or so. I want you to do all of them away. I want you to do all of them and submit. All right. The next one, what is the term for different versions of the same gene? I've answered it before. Alternative forms of gene. What do we call them? The outward physical appearance of a trait is called, did anybody say this one? You mean which? Which of the following genotype represent a heterozygous condition? I've talked about homozygous and heterozygous. Is it capital letter TT, small letter, capital letter T, small letter T, or small letter TT? These are very, very simple question. Gregor Mendel used which plant to study heredity? Told you there, right? So just pause through it and be going through them one after the other like that. The law of segregation states that one allele is always dominant. No, genes for different traits are inherited together. Is that true? Each parent passes one allele for a trait. Then traits blend in offspring. You should go to the this and look at what we did there. Then the gene responsible for determining the sex of a child is located on which of the chromosomes? Autosome, S chromosome, Y chromosome, mitochondrial DNA. The gene responsible for determining the sex of a baby located, is it in the S chromosome or in the Y chromosome? Of course, it is the Y chromosome. If you don't give out the Y chromosome, I don't already told the answer. A trait that is expressed only when two identical alleles are present is called only when two identical are present. That is when you express them. For instance, TT. Go and look at it. What causes sickle cell anemia at the molecular level? Told you that sickle cell anemia is when one amino acid is substituted for the other. Are you getting it? That's what causes sickle cell anemia. All right. Now, the mutation in sickle cell anemia affects which protein? What protein did we say is affected in sickle cell anemia? Is it insulin, keratin, hemoglobin, or collagen? Right? Take note of it. Sickle cell anemia is inherited in what pattern? A, X-linked dominant. B, autosomal recessive. C, autosomal dominant. And D, codominant. How do you get sickle cell? Look at it. AS and AS. If these two people marry, what do you say will happen? There's tendency of them giving birth to an what? A person. There's tendency of them giving birth to what? AS person. There's tendency of them giving birth to another AS person. And there's tendency of them giving birth to what? XS. Are you getting it? Now, when do we say the person has sickle cell anemia? It's when you have 2X. Is he in the X chromosome? Is he in the sex chromosome? We say we have sickle cell anemia. No. So it is not sex-linked. It has to be autosomal. It has to do with the other genes in the body. Let me answer this question for you. It has to be with the other genes, or it has to do with the other genes in the body, not the one for sex-related, like those sex chromosomes that XS and XY. So it is autosomal. Now, is it dominant? It is only dominant when only one, if one is present, and then it is being expressed. We say if it must have the two traits before it is being expressed, we say it is what? Recessive. For instance, sickle cell anemia, you must have the two XS, like XS, before you can actually say this person is what? It's a sickle, or he has sickle cell anemia. Do you get it? So we say a situation where you must have the two allele before it is being expressed, you call that what? Recessive. So the correct answer to this question is autosomal what? Recessive. Do you understand? Autosomal recessive. Autosomal in the sense that sickle cell anemia is not linked to sex. It's not sex related. It's not sex linked. So it is not present in the XY chromosome or in the XX chromosome. We say aside from these two chromosomes, these two, the rest pairs are autosomal. So it has to do with the other genes in the body. That is why we say it is autosomal. Recessive in the sense that you must have the two allele XX before you know that this person is expressing the symptoms of sickle cell anemia. So the correct answer is autosomal recessive. Let us assume if you are AS, then you are sickle cell. You know this S is the trait for sickle cell. The X here is the trait for sickle cell. And if you have only one and then it is expressing itself, you say it is autosomal what? Dominant. That is only one of the trait allele is there, but it's expressing itself. But when you must have two of the allele before it expresses itself, then it is what? Recessive. What shape do red blood cells take in individual with sickle cell disease? Is it round, oval, star shape, or crescent shape? With individuals or in individual with sickle cell anemia, what shape? I told you that is something like this. Exactly. Then the next one. Which of the following genotype represent a carrier of sickle cell anemia? Now, for sickle cell anemia, you know all this AS we are talking about AS in which of the protein is in the hemoglobin. Are you getting it? So you say hemoglobin A and hemoglobin X. If somebody has hemoglobin A and hemoglobin X, is a carrier, but is not expressing it. If you have hemoglobin A and hemoglobin A, you are not a carrier. If you have hemoglobin X and hemoglobin X, then you are a sickler. The full way of writing this AS, SS, and so on is HbA, HB, telling you that it affects the what? Hemoglobin. So this HbA, HbS is seen as what? AS. HbA, HbA is seen as what? AA. HbS, HbS is seen as what? XS. What this one is telling you is that it is affecting the what? Hemoglobin. Is that okay? So who is a carrier? A carrier is somebody who has the sickle cell allele or gene, but is not expressing it. For instance, AS is a carrier. So which of them do you think is the correct answer here? Look at it and choose your answer. Not HBC. There's nothing like HBC or you say na carrier. No. One benefit of I didn't actually talk about this. See, people that have sickle cell anemia, SS, they're not the people they sick from malaria. Their body is resistant to what? Malaria. The mechanism or the pathophysiology of that is something that you are going to learn later. Are you getting it now? So one benefit of carrying sickle cell gene is resistance to what? Malaria. People that have sickle cell gene, they are resistant to malaria. Not tuberculosis, so not HIV, not typhoid. They are resistant to malaria. So you answer the other ones. The ones I know that they are quite. Hemophilia is caused by a defect in what? Hemophilia. Is it white blood cells, clotting factor, hormones, digestive enzyme? Get the correct answer there. Hemophilia is mostly commonly inherited through what? Which pattern? Now, say hemophilia is commonly inherited through, is it autosomal? No. We say hemophilia has to do with what? Sex linked. So it is X-linked recessive. Now, why do we say it's recessive? You may now want to ask, is it not only one of the traits that carries it? Because we say the trait is recessive is when the two of them. No. The thing is that for hemophilia, the only chromosome that actually has this hemophilia is the X chromosome. Are you getting it now? Is the S chromosome. So when the S chromosome, which is the only one present there, has it, and it's expressing itself, then we say it is what? Recessive. So the correct answer is S-linked recessive. All right? Now, which sex is most likely to be affected by hemophilia, men or women? I've said it in this class, that for women, answer it. I have the urge to answer the questions, but A female carrier of hemophilia has the genotype. I've taught you about carriers. H means normal. This person is normal. XS is normal. This one is hemophilic. This one is a carrier. This person is hemophilic. Is that okay? Why is he a carrier? It has one normal, one hemophilic trait. When you have one normal, one hemophilic trait, then you are a carrier. Then if you have two normal, you are normal. Then if you have, for this particular one, for the male, is hemophilic. I believe that we've come to the end of this lecture. Thank you very much for your time. We'll see you in the next one. Do well to submit your assignment on Monday, first thing on Monday morning. No submission, no class for you. Have a nice day. Bye-bye.